So it went out of fashion, and it kind of had some ideas that had to come back into fashion. Now, at the same time, as history moved forward, we're now in the 1700s, right? So we've gone systematically from the 1500s to the 1600s, and now the 1700s. Okay. We're going to talk about moving away from some of the psychology and philosophy at this point to now looking into some of the biological developments that were happening, because we are in a class of biological psychology, and we need to find a way to weave those two words together, biological and psychology. So how does this stuff even combine to create our field and to create our class that we're going to spend time talking about this for? Okay, this guy Luigi Galvani. Okay, just by the name of it, you have to assume he's Italian, right? I wonder if he has a brother named Mario. Should be pretty cool. So anyways, one of the things that he made a huge contribution to was discovering and showing us the demonstrations that helped prove Descartes wrong. Descartes said nerves are moving fluid and hydraulic mechanical forces, right? He began to show us instead that our muscles and nerve cells were producing electricity. Okay? That's a huge thing. First of all, we didn't even know electricity was around for most of human history, right? Now we're starting to get these glimmers of electricity, which is pretty wild, right? Think about, like, you're those guys. You're one of the wealthier upper crusters. You're pooping in a bucket, which is better than the latrine that the non-upper crusters are pooping in, right? Like, that's your big technology that you have is a bucket. Okay? Like, you know, contextually, keep this in mind, because they're making some pretty profound discoveries, and then finding that there's this weird thing, but kind of like a little mini lightning bolt that goes through. Like, that's like alien stuff, right? It's really wild and exciting that you get to discover that. And the way he did this in finding this insight was he did what's called he had a preparation of a frog leg, which is a very nice, fancy way of saying he chopped off a frog's leg and he pinned it to a board. Okay? This is our little frog leg over here. You can see this used to be the better half of the frog. Okay? But, you know, in the name of science, we began to work with our little frog fellow. Okay? And what he was able to do is he could find that if he stimulated and sent a little bit of electricity through a wire into the frog leg, well, first of all, let's say he first wanted to see if the frog leg would do anything. So he chopped off the top half of the frog, and he came, and you can take your little poker, little stick, you know, poke the frog leg. What do you think happens? Nothing, right? I hope. Right? Okay, so we have a controls condition. This is a really important control condition. In your theses, in your laboratories, you have to show not just that something happens when you do a manipulation, but that something doesn't happen when you do a different manipulation. Or if you don't show that, you've got nothing. You have no finding, right? You have to show that the lights turn on when you turn the switch, but you also have to show that when you turn the switch the other way, that the lights don't turn on. Okay? So then he now sent an electrical stimulation signal, a little wire, and now he sent that into the frog's leg. But instead of it just hanging there inert, like a dead frog, the leg goes whoop, and that muscle contracts. What do you think he said? Mamma mia. Right? Like, he's probably like, holy crap. Like, you're in there in this technological age in the 1737, or it's probably about 1750-ish in Noah's life, presumably. And you got something dead that you can now make move. It's like Frankenstein. So like, think about these are like contextually, like, that's a big deal. And so we call this animal electricity, or basically what became known as galvanism, right? Galvani. And at the same time, we were discovering things about batteries and storage of charge. So that's what a battery is. You're storing charge. You're able to save an electrical charge for later use. All right? Volta discovered that. That's why we call that measurement in batteries volts. Okay? So we have this whole other end of science happening, right? As much as Descartes is doing his thing, Da Vinci's filling wax in the sheep brains, we have all these little silos of people in France, Italy, England for Thomas Lois. Different time periods, but you're getting these silos of people starting to pull these different pieces together from psychology, philosophy, biology, physics, electricity. Okay? And so we're now in kind of this stage where society is now electric. Okay? And I just always think of like these like prominent Renaissance guys in like the white wigs doing the electric slide or something, because it's electric. And so as we move forward, we'll talk about this guy, Hermann von Helmholtz. We know that fella?
The name's familiar.
I'm sorry?
The name's familiar.
Yeah, he's a big guy in terms of science. Heavy hitter. Obviously, we're in conversation between Descartes, Aristotle, Galen, Willis. I mean, these are heavy Galvani. I mean, these are conversations with Helmholtz, luminaries in human history of science. Okay, so now we bump up about another 100 years. All right, from 1750, now we're about the 1850 or so. And now he's using these electrical signals that people have been slowly developing over about 100 years in through investigations. And he's doing similar electrical stimulation, but now he's not doing it of dead, severed frogs. He's doing it of live, biological beings, in this case, dogs. Okay, so you have a surgical prep of a dog, and you could stimulate part of the dog's brain and showed that it actually moved the limb. So that's a big step forward too, because you're not just sending a jolt of electricity into the frog's leg, right? So if you electrically stimulate a leg, it just shows that leg will move. But think about what's happening now. You're not sending the electrical signal into the leg. You are sending it into a part of the brain that the null hypothesis says doesn't do jack for your arms and legs. Right? Think about what the null is. Why are we stimulating the brain versus the shoulder or the heart? Heart's a better example, right? The heart could be a good candidate for what moves our arms and legs, but instead the null hypothesis is that the brain won't do anything. You stimulate it with some electricity, it's going to go and do nothing. Instead, it will move a limb in a systematic way. A little bit of volt of electricity, arm moves up. Turn electricity off, arm moves down. Turn it on again, up, off, down, up, down, up, on, up, on. That's a systematic way. Heavy discovery, right? Because remember, brain is no longer maybe a radiator, the way that the smart guy Aristotle thought it was. And so that was a key insight because it showed that our nerves are operating by electrical signals. Okay? So that's a big finding that Helmholtz was giving us through the electrical study. However, it also showed us that they are not acting like wires. So nerves are operating by electricity, but they're not operating like wires. How do you know that? How would he know that? Send a lot of electricity in the brain. Arm moves, leg moves, what have you. In the dog. Okay. Raises its paw, lowers the paw. Yeah.
What would it be because if they're all wires, wouldn't the whole body move?
No, I mean that's one possibility, but the idea though, theoretically, is that we have different wires going in different places. So you stimulate the right wire, it gets the right thing moving, not the rest of it. But yeah, I mean you're right at a broader level, but they were working more specifically at that point. And the way that he was able to determine that was what became a critical feature underlying so much of psychology and neuroscience research that we almost take it for granted now. But it's the measurement of time. Why do I say that? Well, the physicists who study the electricity, we know that when you run it through a wire, you start a stopwatch and you finish a stopwatch. So you turn the signal on and you measure how long does it take to turn the the light bulb on the other side of the room. This is before light so much, but you guys get the idea. You can measure the signal arriving from the start or the onset to the offset across a wire. And there's a speed. So it's a super fast speed, right? It's a speed nonetheless. And if you have a sensitive enough watch or measurement device, you can measure what that speed is because it's physics. It's generally constant. The speed through a wire is always the same with that electrical current flow. So the speed of light, the speed of sound, all these things, right? So, when he stimulated the dog's brain, and you measured the response in the dog's paw, or tail, or ear, or whatever appendage you want to think the dog was moving, you could measure the time it took. And what he found was it took slower than the hypothesis said it would take to move across the wire. What does that tell you? It might diminish. It doesn't actually tell you things about the magnitude of the signal diminishing or maintaining. It just tells you that it's moving slower because it's still sufficient enough to elicit the response.
What are you comparing against again? A wire.
As opposed to a wire, right? So when he's finding that the nerves do not behave like wires in how they conduct electricity because they're actually much slower in the conduction of that signal. So you're comparing the speed of transmission of signal. What does it tell you? Bigger insight.
They're made of something else.
Yes,
Conductive.
exactly. Totally. There's a biological substrate, right? It tells us that there's biological signals that are governing our neural activity. It's not just electrical. We are not robots, Descartes. Right? Because think of the implication. What if that wasn't true? What if he's in his laboratory and he's measuring it and it's exactly the speed? You're going, oh shit, I'm a robot. We're robots. Right? Like, and now when you're thinking maybe I'm the only one that's not a robot, maybe everything around here is a robot, Descartes was right. Right? Like, think of that. I mean, that's a really sci-fi discovery if that were to have been the case. You guys see what I'm saying? But that wasn't. But it was also a phenomenal discovery. Like, holy jeez, we're not robots, which is a very good discovery, right? But it also tells us it opens a whole new door of avenue to explore what is this electrical signal that works through biology, right? It's fascinating. And so we have neuroconduction that is biological in nature. That is the big thing. And now we have this field of study that's open for researchers and scientists to explore neural functioning at the intersection of biology and electricity, governing our otherwise mental operations. That's a big deal. So actually I found out just a couple days ago, I guess, 2 days ago. So Hermann von Helmholtz is my, I think, 7th degree academic grandfather. So for you guys who don't know what that means, so people in academics were really giant dorks and nerds. And what you can do is you can find sort of family trees or lineages. You can trace your lineage, who trained you. So my PhD supervisor was Andy Onlinus at UC Davis. My dissertation chair was Steve Luck. And you can find out who is his supervisor. So that would be like my academic grandfather or grandmother or what have you, right? And so because we're giant dorks and nerds, people have found that they have nothing better to do than to create these digitized family trees on the interwebs now. And so you can go there. It's called Psych Tree or Neuro Tree. Like I'm on Neuro Tree. But you can trace people back hundreds of years. So I'm like 3rd degree, 3 degrees away from B.F. Skinner and William James and all these like prominent psychologists. And then if you trace it back a little further, because it's just time beyond the 3rd degrees, take it 7 more steps. I can actually trace it back to this guy. It's pretty cool. So you can do that if you work with people and whoever your supervisors are, you can trace them back. Maybe that's how you should take your supervisors. You just compare their family trees, see where they really come from, their academic genetic stock. You know. And you can go up to them, I was really thinking about joining your lab, but I did a little research and, you're only like 7 or 8 degrees away from Helmholtz, but, Dr. Armadillo is like 5 degrees, so, I thought maybe he could be a better opportunity for me to get my work done. So anyways, that's Helmholtz. And, okay, now we're moving forward, because we got this idea of structure and function localization, okay? And this is still a major issue in the field. And we're going to introduce this guy, Franz Joseph Gall, German guy. And so now we have this idea that you can structurally locate the stimulation of electrical site in a dog's brain, and it moves a physical location of another dog's body movements. So it gives us the idea that the functions of the brain, what it does functionally, it moves the arm, it moves the lip, it moves the leg, what have you, can be ascribed to a specific structure of the brain. Also kind of like a machine, right? Right? There's a part of the car and it does this thing. This other part of the car does this other thing. This other thing that the car does relies on this other third part, right? And so you start to get to this parcellation of the brain of just localizing a structure to a particular function and vice versa. And when you take that to extremes, you find what Francis Gall found. So he's taking this structural localization of structure function, the idea that, and again, this was based actually on patient data too, very similar to what Galen observed in patients, that Thomas Willis observed too, that there are different deficits that you can see in patients who have psychological sickness or neuropsychological damage or injuries that seems to be consistent with where is the injury versus what is the functional loss. Okay? So he's finding these observations, which are not wrong, but he's taking the interpretation of it quite to the extreme, and he's basically saying that, first of all, he advocated the monism view, that there's a united view of mind and brain, but that the brain is not a homogeneous organ, but it can be parcellated to one thing does this other thing, and this other part does this other thing, okay? And so he developed this theory called phrenology, okay? So sounds like some of you guys might have heard of phrenology before, okay? And so he proposed this idea that we have 35 different faculties or types of sort of traits, okay, of emotions and intellect that are housed in specific brain regions, such as the brain area for combativeness, or the brain area for calculation, if you're mathy, or the brain area for love or bad news or what have you. And he was able to identify those by feeling around your skull to feel where are the bumps. And the idea is the more you use it, like a muscle, that little structure of the brain that he says the structure is responsible for function, the more you use that area of combativeness or calculationness, that brain area is going to grow like a muscle, and it's going to lead to a bigger bump on that part of the brain. So if I feel your skull, I can feel where the bumps are, and I can know what part of the brain do you tend to use more, or maybe not so much. I don't know. And he became very prominent because he'd go to all these rich guys' parlors or living rooms and places that people would congregate. He would read their skulls. And he'd tell people their personalities. Basically, was an astrological palm reader, okay, which are also completely non-scientific and just as crappy as Franz Joseph Gall. But he was very popular. So again, this goes to this idea we've introduced. Science goes through fashion abilities, right? And he was a doctor. I mean, the doctor said it. Yeah. I mean, I don't know they have white lab coats then, but you have this authoritative influence. They're smarter, they're more learned than me, and okay, yeah. And so we trust authority, and authority leads us astray sometimes. And so this is an example of his different areas. The spot for sublimity for consciousness is located right here in case any of you guys were wondering. Let's see your veneration, perceivedness, locality, tune. Maybe that's tuna. No, it's gotta be tune.
Can you carry a tune?
Carry a tune. Okay, I obviously can't. And yeah, all sorts of ideality, You guys can check this out. But that's the idea that he would do with phrenology, and it really caught on. It was a big thing, right? You can describe different functions to these different areas. And it's not necessarily wrong. That's wrong. But conceptually right, we have some issues there. So we talked about this idea of electrical elements, that we have nerves kind of sort of parallel as wires, but then we bumped through Franz Joseph Gall. You don't have to know Charles Bell too much, we're not going to get there. But this is another idea of that phrenology, and the bumps on the skull. And he kind of gave a very famous reading to a guy named Napoleon. So Napoleon was the emperor of France at the time. And he led them to many victories in Europe by following one of the rules of French warfare, which is that if you want to win, you find a non-Frenchman to lead you. It's a joke, by the way, because he's Italian. Anyway, the idea was that he gave Napoleon a reading of his skull that Napoleon didn't particularly care for. He was known for being a very arrogant, very self-sure world leader, and probably didn't like some of the things that he was told about his shape of the skull and whatnot. And so Napoleon had the truly good idea of saying, maybe Franz Joseph Gall is full of garbage, and we need to have a scientific way of testing this. This is a pseudoscience. This isn't a scientifically derived theory and model. And so he commissioned real scientists to do real studies to start to test this idea. He said it needs to have a closer scientific view. And the guy he found to do that was a Frenchman named Pierre Flourens. Okay? So Pierre Flourens is this gentleman right there, a very highly regarded scientist at the time, and he was tasked with testing this idea empirically. So instead of just correlations that Gall said, because I could correlate people who have a bump back here as being high in temperamentality, right? That's just a correlation, right? Correlation is not causation. And he said, Flourens, I want you to test beyond correlations. And so Flourens would do ablation studies, which are basically lesions, or you chop out a part of the brain, or you can do a different version of studying this and stimulate the way that Helmholtz had done with the dogs. We can stimulate an area and see what happens, and then you chop it out, an area out, and see what happens. Now you're experimentally manipulating things. You're not just seeing what's correlated with movement, right? He did that primarily in pigeon species, but other animals as too, and his results largely disputed phrenology. Okay, his model that he put forward was basically called aggregate theory or aggregate field theory. And so he said that contrary to Gall, there is not structural localization of structure to function. However, instead, that all brain areas participate in every mental operation. What's the problem then?
Localization is the actual.
Is what?
Localization is real. It's just not the way that Gall submitted it as terminology. So we have areas of the brain associated with speech, but not vision and things like that.
How do you know?
Because we've learned. Yeah. We know
You
now.
just trust is what you said.
Yeah.
So you believe.
Yeah.
Nothing
Nothing is real.
I mean, that sounds like a religion. You just believe it because somebody told you that you trust.
No, scientific review.
Who?
Scientific review.
Who's that?
The process. The scientific review and study.
So somebody else did it and they told you that vision and
You have to trust something. If we don't trust something, we don't. It's hard to kind of live in society. So.
Indeed. Or you can go and do it yourself.
Yeah, or that.
So that's what this guy was doing. But we're going to see, we're going to juxtapose these two models against each other. Everything in the brain does everything. Structuralization, local function. Yes, question.
So this tie into the importance of like replication. What does tie into the importance of replication?
Oh yeah.
Of the same size?
Oh yeah, we definitely need to replicate. And we're going to see some evidence, and we're going to bounce back and forth, because as we're cut to the chase, it turns out to be both. It's true. And that's why it became such a contentious debate. Some of the things that are the most contentious, right? So you believe that there's structuralization or localization, structure, function, language versus visionaries, and every time I ask you about it, you can't give me a good answer on why that's true, other than you just believe it if somebody told you, but yet you're really, really committed to that model. And let's say that I'm really committed to this model. So we both have our data. And we believe our data because we're seeing it with our eyes. And we're really sure it's not wrong. Well, maybe they're both not wrong. Maybe we have this big void between us. We have these really different perspectives because we're seeing data that's true for each of them. And maybe we need to bridge them together and find ways that they can both account for the data. Mostly that's what happens. Question?
Wouldn't that aggregate theory kind of be supported by the fact that, maybe someone who's born without a cerebellum can still do movement?
Exactly. And that's why I was pressing it on the language and the vision things.
Yeah.
Not at a philosophical level, you don't really know that. So you just believe, you just had somebody give you a good score on a test because you gave it back to them. That we read it in the book, which is the same way I learned it, by the way. It's not anything on here, it's the same thing with me. But there's evidence from weird case studies. So there's people who are born without a cerebellum. The cerebellum is supposed to be heavily involved in motor control. It is. These weird cases sometimes, developmentally, your brain learns to reorganize to still do perfectly fine motor control without a cerebellum. You're sitting there looking at the MRI, what the heck? What? It's crazy, but it's really, really fascinating and cool. So if we take a stroll down this aggregate theory lane, okay, I mentioned he was using dogs and pigeons, trying to determine what parts of the brain did, and basically he's saying that all the brain different areas are participating in behavior and that it's impossible to localize things such as memory or language because multiple brain areas are required. Another reason why I was pressing you more deeply about language, because that area of the brain does not exist. There is not a language area of the brain. I'll give you $1 million for anyone who can show it.
Well, there are parts of the brain that associate with, like, we talk about Robert's area. We talk about how we interpret language or.
Oh, you mean interpret language, not language.
Okay, I know what you mean. Okay.
So what you just said is what he just said. Right? Because there are areas that might be associated, but there's multiple brain areas. Because when you say Brodmann's, I say Vermes. And we'll talk about those different areas later on. But if you're familiar with some of those guys' different areas, multiple areas, which means if multiple areas are, then you can't. It's not really localized to the functions of the structure. See what I'm saying? And it's not meant to be confusing. It's meant to show the complexity of the delicate balance between these ways of looking at understanding how the brain is organized. So at the same time, let's take things back to the USA. Okay? You guys like the AMC TV show Hell on Wheels? It is a good show. It's about guys building the railroad, post-Civil War Reconstruction. And we had some contributions that American data is going to start to play to this otherwise European Western civilization story. And we're building railroads, and you got teams of guys who are laying the actual railroad rails. Now, they had to blast their way through certain mountains and all that, and we're going to look at some evidence that came about that really tells holism, or aggregate field theory. Can you guys see this, by the way? Do you need the lights up? Is this okay? Is this okay, or do you need the lights?
It's okay.
It's okay. Sorry. I have two different questions, and my head's moving in different ways, and it's my fault. So, we have this guy named Phineas Gage. Some of you guys might remember this fella. Very famous. Okay, this is a death mask of his head when he died. Okay, common people used to make death masks of people. It wasn't like a creepy thing. It was just a way to memorialize people in his photographs and whatnot. So he was laying railroad ties. This is a railroad tie, and the way you do it is somebody holds a spike and somebody else hits it. So they were going through the mountains. I believe it was Vermont, and they had to use TNT and dynamite, that is, to blast through certain spots to lay the track. And he was holding the spike, and there was an accident because the TNT exploded before they were able to get everybody out. Usually they're like, all right, it's going to go. We're going to get the heck out of Dodge. And it runs out, whatever. But there was an accident. It exploded. What happened is he had that tamping rod, that big, long connection that you hit with the hammer to drive the spike down. And by virtue of that accidental explosion, it went square through his head, blew off the top of his skull, ate through a huge chunk of his brain and his eyeball, and really messed him up. Amazingly, he lived to tell the tale. They all thought he was dead, but he lived to tell the tale. This is a post-mortem. This is still a skull. You can go see it in Harvard's library. You can see it's kind of healed, but it's got this little bump over there. Gall might be interested in that. And here's old Phineas holding the rod that got sent through his head. Pretty remarkable, right?
Wow.
But what was important about the case study in this case, no pun intended, was that it showed critically some insight into localization structure function because he lost that key medial frontal cortex area, and it was associated with profound changes in his personality and some psychological traits like impulse control. So before this, he was kind of like a guy's guy. Everybody liked him on the railroad, pretty gregarious, blue-collar railroad guy, like to go out and have some beers with the buddies after a day working on the tracks, have some laughs. But after he took a rod through his head, people noticed that things changed for him, as they might. And it really changed his personality. Basically, he became a jerk. Swearing a lot of people, wasn't polite. I mean, he did have a rod go through his head. But the idea, though, is that it showed a profound change, that somehow our personality or psychological traits somehow were localized to these structures because the functions change when you damage them. Very profoundly, right? So how many pigs do you need to fly? How many pigs do you need to see fly to know that pigs can fly? Just one, right?
One.
So a case study of one person can be very powerful as a demonstration of proof of principle. And so in this bigger debate between holism and localization, structure, function, this is a very powerful data point at the time. And so it swung the pendulum of scientific fashion back to the other way. And so the Gall and the whole people say, obviously Florenz was wrong, right? And so everybody's got their very swarmy, rich European scientific thing. Like, well, obviously, Gall was wrong. Let me, drink some more tea and have another martini, right? And so you have these pendulums swinging back and forth in this scientific battle royale between these major competing theories and models of brain function. And that went along with Broca's discovery. So Paul Broca was a French physician. He was mentioned earlier for language, okay? And he had a patient who could not produce language. All he could say was his name, which was Tan. Couldn't produce language. And post-mortem, he identified on the post-mortemings after the patients died, you dissect their brain, and you find that he had an area of the brain basically right about here, the left inferior frontal gyrus, that had damage. And exclusively into what's now called Broca's area, the inability to produce language, but he had no trouble understanding language, which was later found by a German physician named Karl Wernicke to do language comprehension, which is why there's not a language part of the brain. Because it turns out it's a network of interacting things, very much like the holistic view of aggregate field theory, right? But this neuropsychological approach of looking at damage to patients, be it Broca's, Wernicke's, Phineas Gage, Galen's patients, Willis's patients, it's still a very useful model. I use it here in some of the patients that we look at. We still look at brain activity differences. Electrically, coming from Helmholtz and Galvani, we can see electrical signals on the scalp related to deficits in different deep internal brain structures. So we'll get to that later, but it's just to exemplify that approach to understanding and asking some of these questions is still a very useful one. We mentioned Wernicke earlier, okay? So you can see this left sort of temporal parietal junction area where Wernicke's area is, and this left inferior frontal cortex area where Broca's patients were. And so we mentioned some of the electrical studies. I'm just going to move real quick here through some of these. It is just showing more things like Helmholtz did, but now in a little bit more characteristic way with movements of limbs of dogs related to electrical stimulation of specific areas of the cerebral cortex, which gave us the idea that there's an area of the cortex that's particularly involved in motor control in a systematic way. Not just your arm now, but if you micro-stimulate a little bit different areas of that tissue that does your arm, a little bit different area will do your thumb, your finger, your bird, your other finger, your fifth finger. Yeah, you guys know the bird. And so we can see if you had sliced the brain here, and you look at it this way, this purple area, just like this purple, will correspond to different parts. Your foot, your hand, lots of areas devoted to your thumbs, your fingers, face. And so we can start to see a structure to function relationship in that capacity. So we're really seeing that pendulum swing in that direction. Okay, Brodmann did the same thing. It's a German guy. Germans are very good with details, right? Only like racist to Germans, but they're very good with details. And he did what's called cytoarchitectonic staining. So he was able to stain different shapes of cells and find that he could code different areas of the brain are filled with different shapes of different kind of cells. It's also another localization of different structures in different areas, right? This pendulum is swinging pretty far to structure or function, almost to Gall again, right? It's like, wow, there's a lot of scientific evidence, right? The science is settled, right? What more do you need to see? Right? It's convincing, right? Like, it's settled. Well, it's over. Move on, guys. Come on. Yeah. But Carl Lashley is who we'll finish off with today. He tested this, and he said, I want to find, fine, good. Okay, localization, structure, function, good. Find me the memory spot in the brain. Find that one spot that does memory. I want to find it. I'm going to name it the Lashley spot. Plant my flag. He spent his whole career trying to do that, chopping out systematically different areas of rat brains and pigeon brains. And it turns out he could not do it. And he magnanimously admitted defeat and said, fine, 1 second. Is there a question?
No.
Okay.
I realized my hand's up.
Okay, that's cool. And he admitted at one point, he's like, I've tried for decades. I can't do this. He was searching for what that structure-function relationship for memory was, and he found that it was how big the damage was, the extent of the damage, the magnitude of it, not the location, that determined the amount of functional loss. And so what he proposed was basically called this idea refuting localization of function, and instead said equipotentiality. Now this is a modern day view of textbooks of things from fMRI, that just like Gall has found all these different spots localized to all these different functions. Looks a lot like our phonology map, doesn't it? Keep that in mind as we move into the modern era with neuroimaging, because it has been called the modern phrenology. There are limitations that we must keep in mind to be responsible scientists. So basically the point of the story is that Gall was wrong, but so was Lashley, sort of. So you had this equipotentiality. The brain is not just equipotential where everything does everything, like Florence said. It's also not just structure and function relationships the way that Gall and other people like that were advocating. In fact, we now realize it works as a series of distributed sort of network processings connecting different nodes of structures to support our broader range of functions. And so we think about it more like network processing. And so we ask less these days about where a function is located and more about how is it integrated across different regions to produce our psychological experiences and behaviors, and figure out how it operates more as a whole. And the mind is no longer our explanation of behavior, but it's rather the source of the phenomenon that we can now study. And so that kind of wraps up today's conversation. I know you guys probably have a process. I'm just trying to hang out for questions or talking to you guys. it, but that's all I have for you today. Thank you for your patience.